To start off, we have the bacteria cell, which, known to its name, is for bacterial cells, prokaryotes. First off, we have the flagella, which is a whip-like structure that enables movement for the cell. And then we have the cytosol, which is colored in yellow, and it is the liquid in which everything is suspended. And then we have the green dots inside of the cytosol, which are the ribosomes. The circular oblong thing is the plasmid, which contains circular DNA separate from the nucleoid, which is made out of genophores, and it's like chromatin but not quite. It contains the cell's primary DNA. And then we have our capsule, which is colored in navy blue, and the capsule can protect bacteria from engulfment by eukaryotes, and it enables better protection against disease. So it enables bacteria to be better at disease. And then, so it enables it to be better at disease as an agent. And then we have the cell membrane, which is in pink, and it is the innermost membrane of the cell, and it provides selectively permeable, and that is the cell membrane. And then colored in turquoise, and between the capsule and the cell membrane is the cell wall, which, similar to plants, also provides structure for the bacterial cell, and it is made out of peptidoglycan. And then on the outside, we have colored in magenta, which are our pili, which attach bacterial cells to specific surfaces or to other cells. And our three things to describe of the function are plasmids, which are the small circular double strands of DNA. Pili, which are these attached bacterial cells to specific surfaces or to other cells, are capsules, which can protect bacteria from ingestion and destruction by white blood cells in particular, and enhances ability to cause disease. And that's our bacteria. All right, moving on to our animal cell. At the bottom here, we have our cell membrane, which is covered in light blue. And next up, we have our Golgi apparatus, covered in purple, mitochondria, colored in turquoise, which is powerhouse of the cell, energy producing ATP. And then here we have our vacuoles, covered in gold, which store anything just like plants do, except they're smaller and not as prevalent as in plants, and they store whatever nutrients that they need. And then we have our peroxisomes, small little circles colored in pink. Our lysosomes up here, D-shaped light pink, as our peroxisomes. And in orange, to the right of the mitochondria, is our rough endoplasmic reticulum. And we know it's rough because ribosomes are attached to it. And in like this burgundy color, we have our cytoskeleton, which provides structure for the cell. It's made out of microtubules and filaments. And right in the center, we have our nucleus, which is composed of a lot of parts. First off, we have our nuclear envelope, which is covered in purple, which basically encases the nucleus. And then we have our yellow nuclear pores, our red nucleolus, and our gray nucleoplasm. So it's like a Russian doll of a thing, because you have your nucleolus within this nucleoplasm, within the nuclear envelope, and it's a nucleus. And then up to the top left in turquoise, we have, light turquoise mint, we have our centriole, and right up here also we have the smooth endoplasmic reticulum, which is unlike the rough ER, in which it has no ribosomes attached to it. And then in navy blue, we have our ribosomes. Now, the nucleolus plays a really vital role in protein synthesis, as it produces ribosomal subunits and rRNA, ribosomal RNA, to send to the rest of the cell. Yeah, so it creates these ribosomal subunits, rRNA, to send to the rest of the cell, and it basically is the center of protein synthesis, because without our nucleolus, we would not have our ribosomes, and without our ribosomes, we would not have our proteins. So in this way, the nucleolus is primary in protein synthesis. Now, our ribosomes, rough ER, and Golgi apparatus are all very crucial in the protein synthesis. Now, this program doesn't allow me to have the entire picture up, so hopefully, all of it's in the lower half. So we have rough ER, which is down here with the blue ribosomes on it, because rough ER has ribosomes attached to it. Our Golgi apparatus, which is purple, right here. And then we have our ribosomes, which are right there as well. So the ribosomes themselves jumpstart protein synthesis from the nucleolus. They make proteins on site. And then these proteins are then transported through vesicles to the rough ER, where they are continuing to be made, genes swapped, genes replicated. And then after this is done, they are transported through vesicle again to the Golgi apparatus, where these proteins are finalized, modified, and transported to their appropriate places within the cell. Next, we have our lysosome, which contains digestive enzymes, and their most important function is to break down excess or worn out cell parts. So they're like trash cans, for example, because they break down waste and take it out and keep the cell healthy in a way. And they may also be used to destroy invading viruses and or bacteria. Lastly, we have our peroxisomes, which are very important in metabolism, as they contain enzymes to oxidize certain molecules found in the cell, which is very important towards metabolism. Last, but certainly not least, we have our plant cell. Now, colored in green on the very outside, we have our cell wall, which contains plasmodesmata, which I will explain later on. And colored in orange is our cell membrane, which we have a cell membrane in bacterial, plant, and animal cells, and the function is basically, at its basic part structure, is the same. It is selectively permeable, and it allows things to go in and out however it needs to for the cell. Now, the main part that our eyes are drawn to in this is the central vacuole, which is covered in blue, and I'll describe that's function later. Then we have our nucleus covered in orange, our nucleolus covered in pink, as the nucleolus is within the nucleus, and surrounding it here is the rough ER, and on the other side is the smooth ER. Now, covered in purple, we have our mitochondria, which are the same as they are in animal cells, which is to convert energy to ATP for cells. And in pink right here, we have our ribosomes. In yellow, we have our small vacuoles. Green, we have chloroplasts, which are very important in photosynthesis. And these two double lines that we see throughout the cell are cytoskeleton, which is microfilaments, microtubules, very important for cell structure. It's true to its name, it's a skeleton of sorts. Now, our cell wall and plasmodesmata. So the cell wall provides support for this plant cell. It protects against any kind of stress, and it allows cells to develop trigger pressure, which is the pressure of cell contents versus cell contents of the cell as a whole. Now, the plasmodesmata is located in the cell wall, and its function is very important. It allows for intercellular communication directly. So this plasmodesmata links to other plasmodesmata in adjacent neighboring plant cells. And it links to cytoplasm, the plasma membranes, and the ER, and it allows for these cells to directly communicate cellularly, which is very important. And this is located in the cell wall. Next, we have our chloroplasts and mitochondria. The chloroplasts convert sunlight energy to food, photosynthesis, in order for the plant to have its nutrients to survive. which is photosynthesis. And the mitochondria take this food that the chloroplasts have synthesized and break it down into ATP, therefore the cell has energy. So the way in which these two organelles work together is simple. The chloroplasts make the food, and the mitochondria uses the food for energy. Now last we have our central vac wall, which stores water and maintains turgor pressure in the plant cell. It pushes contents of cell towards the cell membrane, and it allows plants to take in more light energy for food, and it is the main storage structure.